In this video, we are going to discuss regarding importance and production techniques of blue-green algae fertilizers. See, any biofertilizer including blue-green algae is basically soil-friendly microbial inoculant that are multiplied in controlled conditions which enhances crop yield. So BGA have been found to be an easily available and economically cheap resource material for production and use as a biofertilizer. So we can define biofertilizer as biologically active product or microbial inoculant of bacteria, algae and fungi or in combination, which helps in biological nitrogen fixation for the benefits of the plant. Now, after water, nitrogen is most often the limiting factor for plant growth, and the crop needs heavy dose of nitrogen to maximize yield. For example, wheat, rice and maize needs 20 to 40 kilogram of soil nitrogen per hectare over a period of 3 to 5 months to satisfy the nitrogen requirement of each ton of grain produced. So, to meet such high demand, farmer must either apply inorganic, that is synthetic chemical fertilizers to their land, or they have to rely on biological nitrogen fixation. Adding synthetic fertilizers have negative environmental effects and also their production is very costly. So these fertilizers during their production consume fossil fuel energy. 1 kilogram of nitrogen fertilizer required about 3800 kg of fossil fuel energy. At the same time, many greenhouse gases are released which causes global warming, and to avoid all these evils, use of biofertilizer is must by our farmers. And today agriculture sector plays an important role in the national economy of India. It contributes about 70%, 17.4% of national GDP and provides employment to about 50% of the working population of our country. The non-judicious and blanket use of chemical fertilizers has critically deteriorated the soil and polluted the groundwater. The indiscriminate use of chemical nitrogenous fertilizers like urea, DAP, etc. have made the Indian soil more acidic and sterile. Hence, at present, the focus of Indian farmer is being shifted to the use of blue-green algae biofertilizers, which are most economically and effective source of nitrogen, not having any harmful effect on soil flora and fauna also. So, blue-green algae biofertilizers are having high efficiency of nitrogen supply to crop plants, but at the same time they are very low in cost. So, let's see how these BGA fertilizers are produced. So, the next point is production technique of blue-green algae biofertilizers. See, many species blue-green algae have capacity to fix nitrogen by symbiotic and non-symbiotic methods, and all such species are used in production of blue-green algae biofertilizers. For example, nostoc, anabina, glyocapsa, oscillatoria, plectonema, or many more, etc. Now, the first step to begin is obtaining the pure culture for mass multiplication. Whenever we go for the production of biofertilizers, there are two main steps. First step to obtaining the pure culture, and second one, that is mass multiplication by different methods. Now, after mass multiplication, the dried flakes of blue-green algae packed in polythene bags and stored for further uses. And blue-green algae biofertilizers produced in controlled condition under polyhouse or greenhouse are commercially important. For the production of blue-green algae biofertilizers, let's see how the pure culture is obtained. So, first step, that is to obtain the pure culture. Here, different species are used in production like nostoc, anabina, and glycopsa, etc. About 5 gram of topsoil is taken from paddy field and added into 100 ml of Fox medium in a flask. Then the flask is well shaken and incubated at the room temperature. The elimination of 1500 lux is provided to the culture to increase algal growth. The algal culture is transferred to 10 ml of water in a tube. Shake the tube to separate the algal filaments, and the content is serially diluted, and every dilution drop is again inoculated in Fox medium containing petri dish. Now here there is a need of preparation of Fox solution. So we have to prepare a Fox solution by using required chemicals in a particular concentration, and mix all the chemicals in 100 ml of distilled water and maintain the pH about 7. So obtaining the pure culture is to be examined under microscope, and finally the pure culture is obtained. Once the pure culture is obtained, then go for mass culture, which can be done by different methods. So second step is mass multiplication. So there are different methods of mass multiplication of blue-green algae, like tank method, pit method, field method, and nursery method. Remember, in all the methods mentioned, soil is used as a carrier for producing blue-green algae biofertilizers on large scale. So let's see one by one all the methods of mass multiplication. First method, that is tuff method or tank method. First, take metallic tray made up of zinc or iron of size 6 inches by 3 inches by 9, which are filled with 10 kg of soil. Then spread 200 grams of superphosphate over it. Then pour water up to 5 to 10 centimeter height. Calcium carbonate is added to adjust pH about 7, and spread sawdust over the soil. Sprinkle the starter culture over the soil and keep the tray in sunlight so that algal culture will develop nicely. Water the tray every day. After the sufficient growth of blue-green algae, the soil is allowed to dry, and these dry flakes containing blue-green algae are collected, packed for further use as biofertilizers. Many times permanent cement tanks are used instead of trays to produce a blue-green algae biofertilizers. Next method is a field method or soil method. In this method, the biofertilizers are prepared in an open field of small plots of 40 square meter. The plots are watered up to 15 centimeter height. Then 20 kg superphosphate is added in each plot. After correcting the pH, 240 grams of carbofuran is applied in every plot to control insect and pest. Now the starter culture, that is algal culture, of 5 kg is provided to each plot, and the plot are frequently watered. After 3 to weeks, blue-green algae is developed in all the plots in clear sunny weather. The soil is allowed to dry, and dried soil containing blue-green algae inoculant is further used as biofertilizer. About 30 kg of blue-green algae inoculant is harvested from each plot. After harvesting, the well-dried blue-green algae flakes are packed in gunny bags, and the bags are stored in cool and dry place. And these blue-green algae bags can be preserved for 3 years without losing its efficiency. A 10 kg of blue-green algae inoculant is recommended for 1 hectare of flooded rice, and the dried blue-green algae flakes are introduced to the field after 10 days of transplantation of rice seedlings. The application of blue-green algae to crop is known as algalization. It increases the yield up to 34% in rice. So the field or soil method for production of blue-green algae biofertilizers was introduced as a package of practice in many states of India. And this method is simple, cheaper, and easy to adapt by the farmers. one can continuously harvest algal growth from the same area by reflooding the plot and apply superphosphate and pesticides. Addition of algal inoculum is not required. Next mass multiplication method is pit method. In this method, make the shallow pits under sunlight and line them on inner side with polythene sheets to avoid percolation of water. Fill the pits with soil up to 20 cm and water the pits up to 10 cm height. After measuring the pH, carbofuran is added to the pit to avoid infection from any soil-borne fungi. Spray sawdust over the soil in all pits and now sprinkle the starter culture over the soil in pits. The pits are watered to favor the good growth of blue-green algae, and this soil containing blue-green algae is further used for algalization as a biofertilizer in future. And the last method of mass multiplication is nursery method. In this method, the farmers can produce blue-green algae biofertilizers with seedlings of rice in the nursery. If 320 meters square of land is allotted for nursery, an additional 40 meters square along the side of that plot is reserved for blue-green algae biofertilizers. The procedure is the same as described above in the field method for production of blue-green algae biofertilizers. And by the time the rice seedlings are ready for transplantation, about 15 to 20 kg of blue-green algae inoculant is available in the side plot. So it is sufficient to inoculate 1.5 hectare of field. Thus, the farmers have not to invest additional money for preparation of blue-green inoculant, and both the rice seedlings and blue-green algae inoculant are ready side by side in the same place. But this method has not become that much popular among farmers. Now, demerits of soil as a carrier material for blue-green algae biofertilizers production. So, soil-based inoculum used in all above 4 methods has many disadvantages like poor inoculation loading, heavy contamination and bulky nature of the carrier, that is soil. So these are some demerits of the soil as a carrier material. Now how we can improve it? To avoid the demerits, nowadays improvements are made for easy and fast production of blue-green algae biofertilizers. First, selection of suitable carrier. Secondly, indoor production instead of production in open area. It is highly suitable and best way for blue-green algae production. Thirdly, proper mixing of culture. And fourth, new carrier materials like paddy straw, rice husk, coconut coir, sugarcane waste and multani mitti are used. The multani mitti is the best and widely used carrier at present for production of blue-green algae biofertilizers. In this culture are developed within 6 to 10 days, and the inoculums must have tar value of 10 raised to 4 CFUG and shelf life of 2 years. So this is all about the mass multiplication and of blue-green algae biofertilizers. Now, how these blue-green algae biofertilizers are applied in farms. So, the methods for the application of blue-green algae biofertilizers slightly differ from crop to crop, and usually one packet, that is 500 gram of packet, ready to use multani mitti based BGA biofertilizers is recommended for 1 acre of rice growing area. The packet is opened and mixed with 4 kg of dried and sieved farm soil, and this mixture is broadcasted on standing water 3 to 6 days after transplanting of rice seedling. And sometimes excess algal material is used to accelerate the algal growth in the crop. And the field should be kept waterlogged for 10 to 12 days after inoculation of blue-green algae to favor good blue-green algae growth. When nitrogenous chemical fertilizers are to be used, then reduce the dose to 1/3 of the regular dose. If fertilizers or pesticides or weedicides are to be used, then keep a gap of 3 to 4 days and then apply blue-green algae biofertilizers. Application of phosphatic fertilizers is helpful to enhance the algal growth, which stimulate faster multiplication of blue-green algae. So that's it regarding production of blue-green algae biofertilizers.